Thank you, Shiva. Welcome everyone to today's webinar organized by Bioingene. Bioingene is a nonprofit organization created to promote plant science research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope that future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We have received a huge response of more than 2,500 registrations for today's webinar. We welcome the audience and thank them for their interest in Bioingene. It is our request to all viewers to please hold all questions for the question and answer round at the end of the presentation. You can type in your questions related to today's talk in the YouTube comment box. You can apply for a certificate of participation via the feedback link and password, which will be provided after the presentation. Submission of the feedback form is mandatory in order to receive the certificate. The topic for today's webinar is Mainstreaming Underutilized Crops, the Story of Foxtail Millet by Dr. Manoj Prasad. Dr. Prasad is working as a senior scientist, professor, and J.C. Bose National Fellow at the National Institute of Plant Genomic Research, NIPGR, in New Delhi, India. He received his PhD degree from the Department of Botany in the University of Calcutta in 1999. Prior to working at NIPGR, Dr. Prasad served as an Alexander von Humboldt Fellow from 2000 to 2003 at IPK in Gatersleben, Germany. His research interests include plant molecular genetics, genomics, and stress biology. Dr. Prasad has published more than 140 research papers, reviews, and book chapters in reputable journals. He is also serving as an associate editor in renowned international journals of high repute, Scientific Reports, BMC Plant Biology, +1, Plant Cell Reports, Functional and Integrative Genomics, Plant Molecular Biology Reporter, Plant Breeding, Journals of Genetics, etc. He is the recipient of many prestigious awards like the Alexander von Humboldt Fellow in Bonn, Germany, the Young Scientist Award, the NAAS Associateship of NAAS, New Delhi, India, the H.C. Memorial Award of the Indian Science Congress Association, the National Bioscience Award of the Department of Biotechnology from the Government of India, the Tata Innovation Fellow of the Department of Biotechnology, Government of India, the J.C. Bose National Fellow of the Department of Science and Technology, Government of India, the Golden Peacock Award of the International Buckwheat Research Association, Slovenia, and the Fellow of 3 national academies, INSA, New Delhi, NASI, Allahabad, and NAAS, New Delhi. We are truly honored to have him with us today. Thank you, sir, so much for being here. I would now like to request that you share your screen and begin your talk whenever you're ready.
Thank you, Liberty, for introduction. Sir, you can again share your screen.
Okay. You can see this?
Yes, yes, sir.
Yeah?
Fine. Yes.
Okay.
Yes, it's okay, and it's already full screen. Okay, sir.
Okay. So, good morning, everyone. Thank you, Kelvin, for my introduction. And first of all, I would like to thank the organizer, especially Subhabroto, for this invitation in the webinar series. And as you know, the nation or the entire world is facing such a difficult situation due to the pandemic spread. But we proved that our academia is unstoppable, and we keep organizing such online programs to keep the scientific community engaged in active research and providing research updates. I appreciate the participants also who are gathered in large number to prove that nothing can stop us from learning, and I hope this webinar series provides you an updated information on the roadmap for outcome-based education and research. Today, my topic has relevance to the ongoing issue of hunger and malnutrition. However, I would be talking more on the genomics part of crop improvement with Foxtail millet as an example. I am moving further onto the topic that is here. You can see mainstreaming underutilized crops, the story of Foxtail millet. So, as you know, the humans are the last one to evolve on this earth, but we are leading the game in terms of numbers. As of yesterday, this time our population count was 7.7 billion, and expert predicts that it will reach 10 billion by the year 2020. To this population, we have assigned three basic things as their basic needs, namely food, shelter, and clothes. We have been told that without these three needs, survival of the human is not easy. But if you ask whether all the 7.7 billion gets the three basic needs, then the answer is no. 821 million are still hungry. For every 10 seconds, a child dies from hunger-related causes, and 1 in 4 of the world's children suffer from malnutrition, which altogether means that getting food alone will not serve the purpose, but getting nutritious food will. If you see the Global Hunger Index data of 2019, the Africas and South Asia, including India, are still in the danger zone of hunger, irrespective of several steps being taken, including the Green Revolution. Adding to this, the Global Report of Food Crisis for the year 2020 shows that 135 million people living in 55 different countries are facing acute food insecurity. This report was released before the incidence of COVID-19, and it is expected that the number would have been now increased due to several reasons, including lack of income and economic constraint among the vulnerable population. This slide shows the statistics on the effect of food insecurity among children. The Joint Malnutrition estimate that 144 million children under the age of 5 years are stunted. 47 million are too thin for their height, and 38 million children are overweight. If you see the percentage of stunted children, South Asia has more than 30% of the stunted children, that amount to 55.9 million children. In case of wasted or too thin children, again, we talk with 14.3%, which amount of 25.2 million children. So realizing the grave situation and to build a better world for people at all levels by 2030, the United Nations formulated 17 sustainable goals, among which 3 are more directly related to food. This underlines the severity of the issue we are talking about. When it comes to food, I want to show an important issue. The largest and the most diverse botanical garden in Kew estimated that there are 11 lakh vascular plants. Among these, 7,000 were used as a food by the early humans, which in due course of time shrunk to just 3 crops, namely rice, wheat, and maize. These 2 crops scatter more than 50% of world food. In addition, there are 12 more crops or plants that altogether 5 animal species supplement 75% of the world's food. Now you may understand the question of how to feed 7.7 billion with some 15 species. That is the reason why we say that there is a high demand or time to focus on mainstreaming the underutilized crops for food and nutritional security, and using the next generation genomics tools for the trait improvement in this crop. So if the participants need more information on what I am speaking about, then they may refer to a recent article published in Advances in Genetics last year. Coming to the first part of my talk, that is the mainstreaming the underutilized crops for food and nutritional security. We call the crops as underutilized when they are cultivated in certain regions and consumed by marginal population. These species would be tubers, legumes, leaf vegetables, and minor millets. In case of tuber, it could be taro, arrowroot, Indian shoot, granola, purple yam, air potato, elephant foot. In case of legumes, it may be wing bean, jack bean, bambara groundnut, kidney beans, fava bean, lima bean, cowpea, horse gram, rice bean, velvet bean, ajusi bean. In case of small or minor millet, it could be finger millet, foxtail millet, proso millet, little millet, banyan millet, teff, kodo millet, fonio, job steer, guinea grass, and brown top millet. So India is the second largest producer of millet next to China, and area-wise Rajasthan and Maharashtra are the toppers. But it seems that the production-wise Tamil Nadu, Uttarakhand, and Andhra Pradesh are leading the race. If you see the nutritional potential of millets, it clearly shows that millets are rich in protein, fat, ash, etc., as compared to rice and wheat. In this table, you can see, or highlighted in blue color, all this composition in 100 gram of grain for all the millets as compared to rice and wheat. In case of micronutrients, including potassium, calcium, phosphorus, magnesium, sulfur, zinc, iron, boron, manganese, and nickel, millets are proven superior than major cereals. Similarly, like the other the previous slide. So if you want to read more about the nutraceutical potential of millet, please read our paper published in Plant Science. So millets are slowly growing, gaining popularity through, as you can see, the media now speaking about it. Professor Banni Rajan from Tamil Nadu Agriculture University, Madurai campus, who has released a banyan millet variety, MDU1, for cultivation and consumption that was overtaken the cultivation of rice in Madurai district of India. So recognizing the potential of nutritional potential of millets, our central government has declared all the millets as nutraceuticals, and the United Nations had declared the year 2023 as the International Year of Millets. But still, it is miles to go to mainstreaming millets in research and to the plates of everyone people. So coming to the second part of my talk, that I will show how genomics tools can help us to working out one of the minor millet called foxtail millet. So the foxtail millet is similar to rice genome, but maybe it is worked out that it is smaller than rice genome. It's a diploid. Haploid chromosome number is 9. A short life cycle, genetically close to major cereals and bioenergy grasses, and it possesses high degree of adaptation to dry environment. That's why these millets are grown in the semi-arid region. And the most important part of this millet is this, that they have better water use efficiency and nitrogen use efficiency, and they are highly nutritive, as I showed. So if you see this graph, that to grow 1 gram of dry biomass of maize and wheat, you required almost 500 liters of water. And but for foxtail millet or other millet, it is just half the water required for producing millets. So for any crop improvement program, you need a good number of germplasm collection, and then you have to screen this germplasm so that you can find out the diverse population, and also screening should be perfect. So this graph shows that the map shows the drought prone areas from where this foxtail millet accession has been collected, and in addition to this, we also collected from different parts of the world, that is USA, China, Kenya, USSR, now it is Russia, Bangladesh, Ethiopia, Pakistan, Turkey. So we have 170 foxtail millet accession or germplasm that are collected from different parts of India or drought-prone area, as well as from different parts of the world. So to screen for dehydration and salinity tolerance of this 170 foxtail millet accession, we analyze the relative dehydration and the salinity tolerance on the basis of relative water content, lipid peroxidation, electrolytic leakage, and antioxidative enzyme like APX, catalase, and glutathione reductase. This graph shows that we have categorized the 170 accessions into highly tolerant, sensitive, and highly sensitive, and they are grouped here. So by this way, we have identified the most contrasting tolerant and sensitive. So Prasad and IC 403579 was highly tolerant with both the stress, drought and salinity, whereas Lepashi and IC 480117, they are highly susceptible. So this physiological and biochemical data was further confirmed by the expression analysis of these genes like APS, catalase, and glutathione reductase. So based on this, when we started this work at the 2008-9, at that time NGS technology was not there. So we, based on this cDNA subtractive library concept, we have prepared a contrast, as I showed you, that we have identified four actually contrasting genotype or accession of foxtail millet for both the stress, dehydration, and salinity. And we did the comparative transcriptome analysis, and we found that this is the dehydration and salinity, 18 genes which are common between these dehydration and salinity stress tolerance. And total transcript of both the library consists of 182. So you can see here only 18 genes, that is out of 182, but the 10% genes are common between the two stress when dehydration and salinity was imposed while using the same contrasting genotype. So we suggest a distinct mechanism to perceive and respond for salt and dehydration stress condition in foxtail millet. So then I am coming to a story recently developed by one of my PhD students, Dr. Roshan Kumar. Now he has successfully defended his thesis on heat shock protein. And so the title is Identification and Characterization of Stress Responsive Gene Heat shock protein as an example. So in the year 2012, the American group of Benexen and Chinese BGI, Zhang et al., they have sequenced this foxtail millet genome, and this is the description. I'm not going details about the genome sequencing, but how we can utilize the genome sequence information to find out the candidate gene that I want to show you. So from the whole genome sequencing and annotation, we identified that when we compare millet, sorghum, and maize, that 1517 genes, they are millet specific. And out of these 1517 genes, we found through geoannotation, we identified 586 genes were functionally annotated as responsive to water stress. So through this, we pinpointed several candidate genes. One is the heat shock protein, aquaporine, NIP transcription factor, Werke transcription factor, and ADP-RF. So we did the cloning and then the characterization. So coming to the story of heat shock protein, as an example, as we know, there are different kinds of heat shock protein, like heat shock protein 40, 90, 100, small heat shock protein. So these heat shock proteins regulate different aspects of stress defense through protein-protein interaction of the target protein. So when we search the literature, we found that in different classes of HSPs are present, but there is no information available for foxtail millet. So through this HMM profiling and database search, we identified that 20 members for HSP 100, 9 for HSP 90, 27 for HSP 70, 20 for HSP 60, and 37 for small heat shock protein. So we check the expression profiled by the qRT-PCR in different abiotic stresses like heat, drought, cold, and salinity stress. And for that, the same for two contrasting genotypes, which was used for the cDNA substructive live reconstruction, the Prasad and Lepashi was used as a contrasting parent for this QRT-PCR analysis. And out of this 37 gene of 29 expressed differentially in either one or multiple abiotic stress. So you can see here that only one, that is the HSP27, was expressed differentially in all the four stress like dehydration, heat, salinity, and cold. So this we choose as a candidate gene for further analysis. When we check the HSP27 in different stresses like heat, cold,